Good morning, everyone. Thank you so much for taking time out of your morning to come here and join us today at the Baker Institute on stem cell tourism near and far, achieving a compromise for the patient. You all know, either from scientific sources or from reading the media, how important this issue is when we start finding some of our leading politicians and our top athletes traveling abroad for stem cell treatment. And we are very fortunate today to have a panel that is just outstanding in terms of top physicians and a top regulator, bioethicist, and another scientific policy person who are going to speak to us on this topic. So many thanks to Dr. Kirsten Matthews here at the Center for Health and Biosciences for organizing this conference, and to our colleagues at Texas Heart Institute for helping us put this presentation together. Now, we have 4 speakers who are going to speak, and then they're going to be available for questions afterwards. And what I'm going to do is just briefly introduce each of the 4 speakers to you now, and they will come up and speak. Please allow them to speak, and then we'll take questions from all 4 speakers at the end. The first speaker, and I'm not going to read the entire bios because they are so impressive for these 4 different speakers. I'll just give a quick summary. But our first speaker, Dr. James Willerson, who you all know, I'm sure, is the President and Director of Cardiology Research and Co-Director of the Colon Cardiovascular Research Laboratories at the Texas Heart Institute at CHI St. Luke's Health and Baylor St. Luke's Medical Center. And he also served as President of the University of Texas Health Science Center at Houston from 2001 to 2008. Dr. Willerson is just the epitome of the top cardiologist researcher and one of the best physician diagnosticians in the country. He and his colleagues performed seminal research on the use of stem cells to repair hearts and cardiovascular vessels injured by heart attacks. And their research at the Texas Heart Institute was awarded the first FDA-approved clinical trial for the use of adult human stem cells to treat ischemic cardiomyopathies and congestive heart failure in humans. So he is really, we're fortunate to have him here as one of the best experts in the world to talk about this particular issue. Our next speaker is Dr. Joyce Frey-Vasconcells, who is our regulatory expert talking with us. She is the president and regulatory expert at Frey-Vasconcells Consulting LLC, and she comes from really an amazing background, both in terms of industry and science. She served 6 years as a regulatory consultant for PharmaNet, working with industry to help foster product development in the areas of cell therapy, tissues, gene therapy, and tissue-engineered products. And before that, she served more than 12 years as the Food and Drug Administration as the Deputy Director in the Office of Cellular, Tissue, and Gene Therapies at their Center for Biologics Evaluation and Research. So we're looking very much forward to hearing her opinions today. The next speaker will be our own very own Dr. Kirsten Matthews, and she's a fellow in Science and Technology Policy at the Baker Institute. You may know her also from her work at the Baker Institute Science and Technology Policy Program, where she oversees events, conducts policy research, and writes policy reports and briefs. And her research is on the intersection between traditional biomedical research and public policy. And she's published several reports and academic papers on the issue of stem cells in her role here. And last but not least is Anna Iltis, who is also, we like to say, is one of ours. She's a non-resident scholar at the Center for Health and Biosciences at the Baker Institute, and she's also a professor of philosophy at Wake Forest University. She serves on several national institutes of health data safety monitoring boards, and she holds a wide range of editorial positions, including co-editor of the Annal of Bioethics book series and senior associate editor of the Journal of Medicine and Philosophy. So there's our impressive group of panelists, and we will start with the first presentation by Dr. Willerson.
Thank you, Dr. Ho. Dr. Matthews, it's a great privilege and honor to be at Rice anytime, in any capacity, and I'm delighted to be here this morning and with you. I'm going to give you a quick update about our progress in regenerative cardiovascular medicine at the Texas Heart Institute, and remind you that any intervention that becomes accepted for use in humans should go through an evaluation in humans before it's accepted in a double-blind, placebo-controlled, randomized trial. That's a standard of excellence and approval today. I say that realizing and predicting that in the future, the human individual genome will be used to identify patients and place them in certain categories for testing of this intervention or another. But at the moment, this is the way clinical trials should be conducted, and this is the way intervention should be approved. I was asked to speak about the accomplishments of the Texas Heart Institute in cardiovascular stem cell research in 10 minutes. I want to tell you that we formed a team at the Texas Heart Institute over the last 15 years or so. And I want to mention the members of it because no one person's done this work. It is a team effort. Dr. Emerson Perrin is my partner in this work clinically. He's a director of our Clinical Stem Cell Center. He's an interventional cardiologist and one of the best I know. Native Brazilian, trained at the Heart Institute. And he has injected every human heart that we've treated, which now must be in excess of 500. Dr. Doris Taylor has taken various organs, depleted their cell structure, and then restored them to normal function with stem cells. This includes the kidney and the liver and the lung and animal hearts. And so one would have a scaffold in which to create a human heart from stem cells that are added. Dr. Richard Dixon is a molecular biologist, stem cell biologist, who's developed means to attract and retain stem cells in the heart. Dr. Robert Schwartz and our team has taken human skin cells, a biopsy from your hand, and converted those human skin cells into your contracting heart muscle cells in 2 weeks in culture, treating them with 2 genes. And the same for human adipocytes, or fat cells, placed into culture. Our team, Dr. Schwartz, have converted those cells into your contracting heart muscle cells in several weeks in culture. So you can see the background for some of the work that I'm going to tell you about. First, we led the first clinical trial in the world for treating patients with ischemic cardiomyopathies. Your heart is fist-size shaped. If you close your fist and place it over your left chest, that's the size of your own heart. It'll vary, bigger and smaller individuals, men and women. But normally it contracts vigorously and relaxes vigorously. When it's injured, usually by a heart attack, but it can be a virus or drug or aging or genetic predisposition, parts of it don't contract normally, and it starts to contract like this. When that happens over time, it gets progressively larger and fails. It can't serve as a normal pump to pump blood to various organs in the body. Patients who have this end-stage heart failure die within about 3 years. They cannot put their head down flat to sleep. They can't breathe. They sleep sitting up if they sleep at all. They can't walk any distance from here to that wall. They have no energy. Heart and vascular disease is the #1 killer of men, women, and children in our country and throughout the world. More women die of heart and vascular disease than of all cancers combined. And 1 out of 2 men worldwide have heart and vascular disease during their lives. We lose a child on our playing fields in the United States every week of sudden death. Those are all cardiac-related deaths. The patients that we treated in this first trial, which was in Brazil, where we had a collaborative clinical relationship in Rio de Janeiro at their heart hospital, had basketball-sized hearts. The heart had been injured, it had progressively enlarged, patients had heart failure, and they had basketball-sized hearts that just quiver at the top. End-stage heart failure is the most common problem in heart disease. These patients were on a transplant list, but they were never going to get a transplant there. And so we petitioned the Brazilian government to let us treat them with their own bone marrow-derived stem cells injected directly into their hearts with a catheter called a NOGA catheter. This catheter, when it touches the inner wall of the heart, measures the electrical potential there at the site it touches and the velocity of wall motion. It has a needle on the end of it, can protrude and withdraw. So we could inject putative stem cells at each of 15 different sites that were reversibly injured, not scarred and not normal. Within 2 months, the patients that we treated had better blood flow to their hearts. Within 4 months, they had better contraction. And within 4 months, some were jogging on the beach who couldn't walk from here to the wall. You heard that we were awarded the first FDA-approved U.S. trial to treat these patients, same patients, but at the Heart Institute in St. Luke's here. And that work was reported in the American Heart Journal 5 years ago. What we found in that trial was that about 60 years of age, human stem cells are dysfunctional. They're sick, they don't work, and they won't help the failing human heart. But if they're functioning normally, they do help the human heart and improve the blood flow and the function. This was the first such documentation of that, and I am certain that stem cell dysfunction is part of the aging process. The aches and groans, bone problems that we have, failure of various organs, I think is largely a stem cell deficiency. If so, then we'll have a chance to slow down the aging process. We've led a consortium of 5 clinical centers in the largest clinical trial done to date in patients with ischemic cardiomyopathies. This is sponsored by the National Heart, Lung, Blood Institute. And again, we reproduced what we had found before. Stem cells are functionally normal when we'll get an improvement in the function of the heart. And then more recently in Spain, now we're concentrating on specific stem cell types, not just the bone marrow pool. The bone marrow pool helps if the cells are normal, but specific stem cells are better. In this trial, mesenchymal cells, but taken as precursor, early vintage mesenchymal cells from the bone marrow from a 20-year-old donor, a young man. We've also found that humans can exchange their mesenchymal cells without rejection. This is maybe one of the only cells in the body that would be true of. I think there's a retinal cell that also can be exchanged between humans. So we took these mesenchymal cells from a 19-year-old donor and treated 45 humans with ischemic cardiomyopathies with his cells. We could be certain these cells were functioning normally. We had 15 controls. And in this trial, which was also published recently in Circulation Research, we showed that the highest dose of these cells prevented death in a 3-year follow-up in these patients and improved the function and blood flow to the hearts. We're very excited about that. And then another multi-center, multinational trial using adipocytes, liposuction, and the mesenchymal cells taken from the liposuction tissue and treating these patients with ischemic cardiomyopathies, again showing that they improve the blood flow and function of the heart and reduce clinical events. Each place these are published is shown in the bottom of what I'm showing you on the slide. And more recently, we've used an aldehyde dehydrogenase positive stem cell from the bone marrow, it's known as bright cells, to inject into the calves and thighs of patients with severe peripheral vascular disease at risk for amputation of their limb. And I can't say much more about that because we're just now submitting it, and this data is protected at the moment. We will help lead another NHLBI-sponsored multi-center clinical trial in patients with ischemic cardiomyopathies using a combination of 2 different stem cells, which in preclinical studies looks to be better than the single stem cell alone. This includes a resident cardiac stem cell, the c-Kit cell, present in all of our hearts, and the mesenchymal cell. And they'll be compared against each other, and they'll be used together in patients with ischemic cardiomyopathies. This is a 7-center consortium founded by NHLBI. Lessons learned, I've alluded to some of them. Human stem cells are often dysfunctional about 60 years of life. Cardiac stem cells may be injected into human hearts, patients with ischemic cardiomyopathies and other problems safely. When healthy, bone marrow-derived human cardiac stem cells improve heart function and coronary blood flow modestly in patients with ischemic cardiomyopathy. Precursor allogeneic from another person. Mesenchymal stem cells taken from healthy, youthful donors improve cardiac function in subsequent clinical course in patients with ischemic cardiomyopathies during a 3-year follow-up. Adipose tissue-derived human stem cells improve function and blood flow in patients with ischemic cardiomyopathies. And I've already pointed out number 6. Number 7, preclinical studies suggest combinations of cells may be better than single cells alone, specifically the c-Kit and mesenchymal cell. And we're very excited about another finding. We have found, others have as well, that our hearts have a stop growth pathway that inhibits the further growth of your heart and mine after 2 weeks of life. We were not intended to regenerate our heart to any significant degree. The salamander's different. Cut his tail off at any time and he'll completely regenerate it. This stop growth pathway has the unlikely name of a hippo pathway. It's a kinase cascade. A member of our team, Jim Martin, has shown that inhibiting this stop growth pathway by knocking it out allows a murine heart to completely regenerate the area of injury after an experimental heart attack. Whether we knock it out prior to the heart attack or 3 weeks into the heart attack with the animal with significant heart failure. We're very excited about this, and several of these papers have been published recently in Nature, and we have moved into a larger animal model. But if this is safe and works in humans, it's a game changer. This is a stimulation of existent myocytes. It's cardiomyogenesis rather than a stem cell phenomenon. So there's probably a place for stimulating the intrinsic capacity, inhibiting the inhibition of the intrinsic capacity of the heart to regenerate itself. Why do we have a stop growth pathway? I don't know. But probably because our Maker didn't intend the heart to enlarge in perpetuity, but only to reach a certain size. We believe this is a property of every species on the planet, except the salamander and maybe the zebrafish. I've mentioned Dr. Schwartz and our team's contributions. We have also restored aged mesenchymal cells to normal youthful function in vitro and in vivo by treating them with factors that overexpress telomerase and myocardium. Potential for our return to the fountain of youth? Maybe. The future, from my point of view, the use of allogeneic, youthful, human cardiac stem cells, combinations of cardiac stem cells. We've got many different stem cells in our body. They're there for a reason. It would have been logical to assume that combinations would be better than one alone, and I'm confident that's the case. Develop methods to stimulate intrinsic cardiac regeneration after injury, inhibiting the stop growth pathway in humans, and evaluate derived cardiac stem cells created from human skin cells, fibroblasts, and adipocytes in cardiac repair. We're fortunate to be close to Rice at the Texas Heart Institute. We're partners with Rice, and we're very proud of that. Thank you.
Well, first of all, thank you. It's a pleasure to be here this morning. I have a few slides, so I'll go through them very quickly, but the organizers have my slides, so if anybody wants them, you can just request them. All right. In 2005, FDA issued 3 rules that dictated the regulatory authority for human cells and tissue-based products. And this outlines those 3 rules. I'm going to talk mostly about the first one, the establishment, registration, and listing. That's the one that came out first. It describes when you are regulated by FDA and when you can go directly to market. And this is the criteria you have to use. You have to answer these 4 questions. If you're minimally manipulated, which I will talk more about and what that means. If you're using the cells or tissues for homologous use. Do you combine it with a drug or device? There's a few exceptions. Or if the cells have a systemic effect or is dependent upon the metabolic activity of the cells. And you can follow this algorithm to decide whether you're a tissue or a therapeutic. As any company that's developing this, it's your responsibility to decide which pathway you follow. There's no requirement to go to the FDA to make this determination. However, if FDA finds out you're doing something that for some reason you have associated down the wrong pathway, they will let you know. Maybe not in a real timely manner, but they do let you know. So I'm going to talk a little bit about minimal manipulation. This has been a really hot button. In the tissue regulation, the registration regulation, it says they separate it out into 2 things, structural tissue and cells or non-structural tissue. And structural tissue is determined as processing that does not alter the original relevant characteristics of the tissue relating to the tissue's utility for reconstruction, repair, and replacement. Okay, you can also all see that there's a number of terms here that are can dictate which way, whether you're a structural tissue or not. Cells and non-structural tissues are processing that it does not alter the relevant biological characteristics of cells and tissues. I would say the biggest controversy right now is things that are regulated based on structural tissues. In 2014, FDA issued the guidance document trying to clarify what they meant by minimal manipulation. And this year, actually this month, FDA held 2 meetings to discuss the merits of 4 guidance documents they have issued. One is the minimal manipulation. One is how they're interpreting homologous use. They also issued a guidance on adipose-derived stem tissues, and then guidance on same surgical procedure exemption. And September 8th of this month, they had a meeting to discuss the scientific evidence and the developments of these types of products. Then the next week, the 12th and 13th, they had a Part 15 hearing to request for comments on the draft guidance documents. Now, I don't know how many people know about what a Part 15 hearing is, but basically, that's a hearing where it's open to the public comes and gives their opinion, and FDA has to sit there and listen to you. They don't say anything, which I think there's a misconception. So if you ever hear about a Part 15 hearing, don't expect FDA to say anything, because they're not. Now, the docket on all 4 of these documents is open till the 27th of this month. So if you want to comment, you have to comment by then. FDA will then review all the comments that are submitted and will adjust the documents as appropriate. So the scientific meeting, I'll go a little bit about what these are. Clearly, they determined that there's not enough known about the science around stem cells. The manufacturing changes can have a significant impact on the functions of cells. And they had both scientific people there as well as industry. They also had 2 individuals, 2 physicians, that presented case studies of people who had been given stem cells inappropriately. And the first one was 3 women in Florida who had age-related macular degeneration. Now, they had just been diagnosed with it, so they could clearly see. They were still driving, but they decided they wanted to try and stop this disease. So there was a clinic. Actually, this clinic was on clinicaltrials.gov, so fair warning, it turns out, even I did not know this till 3 weeks ago, that not every trial on clinicaltrials.gov is actually overseen by regulatory agencies. And these women had found this trial, supposed trial. They were injected with their own adipose-derived stem cells in both eyes, which FDA never allows you to do that, first of all. 2 to 3 days later, they were in extreme pain. So they went to one of the university emergency medical centers, and they looked at these women's eyes. Their lenses had detached from their eye. It appeared that, well, like the physician who was saying, he said, it's like what we do in cataract surgery when we inject an enzyme to dissolve the lens. That's what was happening to these women. In 1 week, both of their retinas had detached, and they were totally blind, when they could have lasted a number of years with some eyesight. Then the story that probably hit the news, I'm sure a lot of you have seen it, was a man who had a stroke. He received actually several stem cell injections, but in China, South America, and in Mexico. In Mexico, he actually received 2. One was his own stem cells as well as a neural-derived stem cell. He got back to New York, after 6 months, started experiencing extreme back pain. He went to the local hospital, one of the big university hospitals. They did an MRI, and he had this massive tumor that was about this long in the lower part of his spine. They looked at it, they opened him up to do surgery, they looked at it, and it's just this gelatinous mess. They can't even take it out. It's not a solid tumor. When they did the diagnosis of this tumor, number one, it's not his cells. They have no idea what cells these are, and it fits no definition of any tumor known to any physician in the US. They have no idea what this thing is, and it is extremely aggressive. They treated it with radiation therapy, and that helped for a while, and it's now growing back. He will probably die from this tumor. So, you know, stem cells, even if it's your own stem cells, is not always safe. It depends upon what people do with those cells once they come out of you. So in the Part 15 hearing, comments varied, all the way from FDA, you need to regulate this field more, to people saying, we want FDA out of this. You have no business being here. Most comments, I have to be honest with you, centered around the issue of adipose-derived stem cells. I would say most of the illegal clinics that are out there, that's what they're doing. Some indicated that FDA should not regulate autologous cells. I just gave you 2 cases where just because it's autologous does not make it safe. So minimal manipulation. So what do you need first to determine whether you're minimally manipulated? Well, FDA's guidance document says you need to determine the main function of that tissue in the donor. Yeah. Okay, FDA made a mistake by introducing a new term, main function. Don't really know what they mean by that. You need to think about what is the tissue that's being collected. So for example, FDA's interpretation is for adipose-derived stem cells, you need to harvest fat from the donor. So the main function of fat is not the cells themselves, but it's to cushion. So there was a lot of issues around that one. Definition, physically support or serve as a barrier or conduit. So cells aren't going to do that. Manipulated, have you altered the relevant characteristics relating to the tissue's utility? So what's original characteristics? Well, is it present in the tissue of the donor? Relevant, does the characteristic have a meaningful bearing on the tissue's utility for reconstruction, repair, and replacement? That is, the strength, the flexibility, cushioning, covering. So cells isolated from fat obviously don't have that cushioning capability. It's all of the other structures in fat that do that. So that was a critique that most people felt FDA was limiting the original characteristics of fat, because even the cells in fat have important functions. And people felt FDA was dismissing that. Cells or non-structural tissues, it's a little bit easier. Basically, it's if you do any processing, you're basically manipulating the cells. And because FDA has learned, and science has shown us, that when you manipulate cells, you can change their differentiation capacity, their activation state, their proliferation potential, and even their metabolic activity, which can have really serious consequences. They give examples in the guidance document as to what they mean by minimal manipulation versus more. So the bottom line is, for cells, if you culture them in any way, shape, or form, you're more than minimal manipulation. You're automatically regulated. But like I say, the critique for most people was, again, the adipose-derived stem cells. And they said the stem cells in adipose-derived provide endocrine properties in the fat. So upon isolation and use for inflammatory conditions, this is performing the same basic function, and therefore they should not be regulated. We'll see what FDA does with this. So what happens when FDA finds out that you're doing something? They get reports from all kinds. They get reports from patients. They get reports from competitors. They get reports from physicians. They get reports from media people. So when this happens, as a rule, FDA will send inspectors in to find out what you're doing. Now, I have to admit, they're not going to shut you down right away. They're going to tell you, they're going to evaluate what you're doing. They will inform you whether they feel you're regulated and that you need to submit an IND if you are regulated. They will even give you the person's point of contact on how you can work with FDA to get the IND submitted. They will give you an opportunity to comply with submission. FDA, however, will come back in to see if you have complied. They will check up on you. If you don't comply, a number of things can happen. They can issue an untitled letter. If this keeps going on, they'll issue you a warning letter. After they issue a warning letter, they will continue to monitor as they put their legal action together. FDA does not move swiftly unless someone dies. Then they will come and shut you down. They will try their best to put a legal case together, and that legal action in the end can result in seizure of material. It can result in injunctions where FDA has oversight and you can't do anything without FDA approval. Other times, and I'll give you a case in a moment, where there was a permanent injunction and they had to cease operations in the US. And then the last is they can do criminal penalties. So the first example is Regenerative Science. In 2008, FDA informed the company that they needed an IND. In 2010, FDA sued the company to stop performing the procedure. So as you can see, it took 2 years. FDA was constantly going in and looking. This company was using, I think, bone marrow cells. In 2014, there was a final ruling by the United States Court of Appeals that upheld the lower court's ruling that said the company had violated federal laws regulating the manufacture and labeling of drugs and biological products by producing, as part of what they claimed their medical practice, a substance consisting of a mixture of patient stem cells and an antibiotic. They issued a permanent injunction against the company. So what's happened since then? The company's now called Regenix. They have 2 stem cell products if you go to this website. They have a cultured product that they clearly state is only available in the Cayman Islands. They have another product that's the same-day stem cell. It's available in the US and internationally. What I could find out about this is it's a bone marrow aspirate mixed with growth factors from autologous blood platelets. It's not clear to me yet. I think this is probably also regulated. But we'll see once what FDA does with this one. Celtex. A lot of you should know this. It comes from Texas. Their business was to separate, multiply, and store adult adipose-derived mesenchymal stem cells for a variety of uses. FDA found out about this when your former governor went to the press and said he had had a stem cell injection into his bag. So guess what FDA did? They inspected, found many violations. If you read the letters on this, the warning letter on this, it's actually pretty scary violations that they were doing. FDA issued a warning letter saying they were more than minimally manipulated and it was non-homologous use. They were treating many, many different diseases. They wanted them to correct the violations and told them they needed an IND. They continued to operate. To this day, they operate. They advertise on their website that they can treat these variety of diseases. They claim that there's well-documented trials around the world that says MSCs slow or arrest progression of the conditions. So I looked at their articles under a variety of these conditions. None of them describe clinical benefit, and in most cases, there's no human clinical data. It's all data based on mice and rats. And I can tell you from my experience of years in the regulatory agency, mice and rats rarely predict what's going to happen in you. They claim they're conducting clinical trials. They indicate that the clinical trials, if you go on clinicaltrials.web, there's no clinical trials listed by this company. There's no indication of the clinical trial sites on their website, and there's no indication that there's been regulatory oversight. So you kind of have to wonder what clinical trials they actually are conducting. However, they did say that the results will be published shortly. If you enroll in their clinical trials, you actually have to pay $6,500 just to enroll. Then you have to pay an additional $4,000 to $13,000 for administration of the product. First of all, FDA doesn't ask you to pay to enroll in a clinical trial. And number two, unless you have FDA approval, you're not allowed to charge patients for the therapeutic during a clinical trial. So, and in addition, you have to travel to Cancun, Mexico for treatment. So it's clearly not a clinical trial that FDA is regulating. So in summary, there's many issues that surround the regulation of cells and tissues. I understand, and so does FDA, that patients want the right to their own cells. Once cells are isolated, many things can be done that may not be safe. So just because it's autologous doesn't mean that it's going to be safe. Scientists, regulators, doctors, and patients need to understand that bad things can happen without full understanding of what has been done to the cells and proper controls. Is there a more risk-based approach that could be used? Yes. I was there at the FDA when we instituted these regulations. There were several things that I fought. I thought it was too blanket. They claim it's risk-based. I personally think it's a line in the sand. And so I agree with you. I think FDA does need to have a little bit more flexibility. So I'm not sure what FDA is going to do with these divergent responses that they have received. But there's clearly no easy answer to this. Thank you. If you want my contact information, there it is.
I feel a bit intimidated coming after these two distinguished presentations. I'm actually not going to use slides. I'm just going to talk a little bit. My main idea is actually to talk about two areas. I'm going to talk a little bit more about this area of unproven stem cell interventions, about the U.S. and abroad, what's going on, a little bit more depth than what our previous speaker talked about. And then I'm going to talk a little bit about some proposed policy options that were discussed, I think, last week in the hearings, and overall some of the things that are going on in different countries. So as we can see, this unregulated stem cell-based intervention space is actually pretty controversial. There's a lot of promise, and I think Dr. Willison pointed out in his slides, a lot of promise in this area, and I think that's why everyone's really excited about it. But this unregulated space could actually hurt, I think, the science. It could hurt our ability to move the science forward. It could make people wary and afraid of using these treatments when we can see that there could be a lot of good, and I believe there will be a lot of good in the future. So taking a step back, I wanted to start by framing what we mean by the unproven stem cell interventions. If you actually did a quick Google search, and I've had my students do this before for stem cell clinics on the web, you will end up with this huge selection of different clinics around the world that give you an array of cures for a kind of a plethora of different diseases. And you'll see things which kind of disturbed me, and the students as well when they did this study. They found there were cures for HIV/AIDS being promoted, autism, and I don't really feel like I truly understand what causes autism, and how stem cells could fix that, multiple sclerosis, diabetes, any kind of disease like that. You just had this long menu for the exact same procedure was going to cure everything. This is also kind of known as stem cell tourism because there's a lot of individuals who are actually moving from a more regulated state like the United States to another country that has a little bit less regulation, including like Russia and Thailand, for instance. We've also seen others that advertise stem cells less for medical use, but for cosmetic and anti-aging property. They're looking at making you feel and look younger. The students found this also kind of hilarious because we found a website that specifically had a before and after shot. And the before shot, it was a Chinese clinic, was a Chinese woman who looked like she's in maybe her 50s. And the after shot looks like a Caucasian blonde on a beach with a tan. And we were like, wait, what? So I'm not really sure, but it's one of my favorite ones, so I've used it for years. I'm pretty sure they pulled it offline, but I just couldn't help myself extra putting it up there. As indicated, most of these clinics' websites will actually not include data, but mostly rely on patient testimonials. They'll talk about the impact on themselves and the effectiveness they saw of the procedure for themselves, or some just showing what they saw the procedure was, maybe the doctors or clinicians showing the procedure. They don't offer a lot of data or link to publications, as she's mentioned. Sometimes they have self-published paper on there, review papers, and clinical data on animals. Very rarely do you actually have anything that comes across as being kind of more of a legitimate science paper on it. Actually, almost always there is not one. So we see these clinics, I guess, in foreign countries, but also in the United States as well, as she mentioned. We saw Celltex and Regenix here, but there's several other that are here in the US. But some of them will actually offshore, as Celltex seems to do now, their procedures to another place, so they'll recruit within the United States and say you can go to Mexico or the Caribbean or some other country to actually get the procedure. Some others do offer the procedure here. They're most commonly focused on pain management, debilitating diseases. They get a lot of different people to kind of be spokespersons. Most often they like to have athletes that tried the procedure, for instance. And then they just give a long list again of different things I think they can cure as well. They do simple treatments, it seems. I think she described it pretty well. Looking at the adipose or fat cells is very, very common. Moving from one location to the other. They're most often autologous, although some might be from other donor sources, even if whether or not they actually state this or not. And again, they offer little to no data to talk about their effectiveness. The pricing is also something of interesting. The patients are going because they're seeking an alternative, but these procedures cost thousands to $10,000s of dollars to actually do, and some of them ask you to go multiple times, so the bill could end up being hundreds of thousands of dollars that you're wasting on it. I've studied this area for quite a bit. I've talked to a lot of individuals who are interested in going to these particular clinics. I've seen patients who've come back with their disease, but also even worse, and then she's pointed out some examples of that with huge side effects. There have also been cases of death, and it looked like the individual in New York was heading that direction, unfortunately. I've also had people who were actually happy with the procedure come back. Scientists have suggested this might be a placebo effect, meaning that it didn't really help them, but they're just feeling good, and they're just excited that somebody is giving them hope and doing something for treatment of their disease. But perhaps there actually might be an effect that's there, I would like to have us all think about. We could argue that, they argue that this is the reason why we shouldn't close down the clinics, because there might be a chance it's helping. But my problem actually comes in, and again, it leads with the previous speakers, the lack of consistency that we're seeing in the data collection. Not just between going to one clinic and going to another clinic, but within a clinic in itself. They don't seem to approach the procedure in really a truly scientific fashion, as Dr. Willison presented. His work was. They aren't checking to determine the consistency and quality of the cells. Sometimes they're not even checking to see if the cells are actually alive. They can often skip that step entirely. Often the clinics don't record the actual dosage of what they're doing. And there's sometimes this idea that the more cells, the better. But I think there's actually some data, clinical trials they've been working with that shows that sometimes that's not true. There could be an inflammatory response where actually less might be more. Most likely, if this is actually going to work, there's probably going to be a range of optimal dose. The clinics aren't really looking for specific health marker outcomes at the end. They're looking at general data, how well the patient feels. You'll see these slides, 1 to 10, how happy are you? And like most of ours said 9. They'll have things like just general after the fact, they felt like they moved better, but not a before and after assessment seen, at least what they show publicly. And they don't tend to wash over the patients long term. It's like a one-time procedure that they'll see them, maybe one follow-up, or if you're going to a different country, no follow-up at all. And the only other follow-ups might be as if they ask for multiple injections. Remembering again that if you're adding a cell, the whole point is for it to stay there for life. You're wanting to actually make an improvement for long term, so therefore this would be something to follow up on decades, not just 1 month later. We don't see any patient testimonials talking about their negative effects. That's not surprising, I suppose. These just aren't presented, they're just the positive effects. In addition, the patients might no longer be eligible to participate in trials that Dr. Willerson's doing, for instance, because they've already started testing and playing with different cells in their body. I'm not sure if he'd be interested in having them go into his, because they might have an impact on how his actual trial is working. And there are these great trials going on, and there's going to be more. And those legitimate trials are now having to fight these clinics coming out with their promises, overpromising the results, charging for patients, while he's telling you what he feels like is going to happen and doing it in an honest way and trying to get good data to prove it. I know the FDA is often portrayed, and I think this is what she hinted at the hearing, as being in a way of cures. But it is really organized to help the public and not prevent the public from these life-saving interventions. I will acknowledge it is a man-made construct. We all made it together as a society. And there, like all these institutions, there's ways for it to change and ways for it to be fixed in the future. So the FDA, I think, is already starting to look at ways to kind of adjust itself, as we saw with this hearing, but in other criticisms as well. There's been a movement for right to try, expanding access, and so the FDA's response has actually been to change its expanded access program as a result. It actually changed the paperwork required to get these clinical trials and moved it down to only 7 documents from what is estimated to be 100 hours of paperwork to only what they estimate to be 1. So we see there's a lot of potential for them to streamline, and I think they're interested in helping as well. One of the proposed regulation changes that's in front of Congress is called the ReGrow Act. That's known as the Reliable and Effective Growth of Regenerative Health Options that Improve Wellness Act. I've been told that there's a little guy in a small office that figures out these acronyms. So I'm going to go with the ReGrow Act because that's much easier for us all to remember, I think. The point of this law that they're considering is actually to look at interventions and give it conditional approval during the phase 3 trial. The idea would be is that you could actually help companies reduce their risk by allowing them to market it a little bit early. And this would also allow maybe patients to get into the actual trial a little bit earlier. It would get more, I think, companies into it, the actual trial itself. They have some regulations on who would actually fit into this particular space, so ones that have lower risk. But it's actually kind of a new and kind of a bold idea. It hasn't been done in the US, this conditional approval, before. And it's kind of off of this model that's going on in Japan that just passed in 2014 and became active law at the beginning of this year, although we haven't actually seen anyone going through it yet. Another proposal that we saw at the hearing was actually an intermediate or middle ground between the no regulation and a full clinical trial. And one of the ideas proposed is actually looking at a stem cell registry, which would look at unregulated processes and require them to give data, their analysis, do more reporting of what they're actually doing in the clinics, including side effects, give the treatment protocol, patient outcomes, give the ability for the FDA to peek at it and maybe have other individuals do assessments. And this could help patients and physicians more effectively identify some sound interventions from what we think isn't. With regard to both options, I think my concern, though, is that if these interventions prove not to be effective, will the public actually allow us to pull them off the market if they've been conditionally approved? And will this give too much burden onto the FDA and end up turning it into a biologics police force of some sense? The FDA is already understaffed, I would say. They don't have the resources right now really to move through the clinical trial application process, I think, as fast as we want. But I don't see in the future the federal government giving large quantities of money for them to hire a huge amount of staff. So this would just add more to their burden. With all these options, though, I do see that there actually is a common idea, which is that they're still trying to work with the FDA instead of around the FDA to find a way to fix it. Perhaps there's a way that the FDA, if it needs more technical advice, there's ways for the scientific community can be better engaged with it. And perhaps if there needs more funding for staff, there's going to be ways that we should look into, and the FDA and government should look into public-private partnerships. But I think overall, and everyone at this table would agree, that safety is really also the common goal everyone has, something that the patients, regulators, and physicians are all interested in. We all want to see stem cells and regenerative medicine live up to their potential, and I believe this way we could accomplish I believe the way we can accomplish this is by helping the scientists and clinicians really move through some kind of a regulation process so that we all feel safer with the product at the end. And if it's broken, then I think that we need to spend our time trying to fix it instead of throwing this regulation away. Thank you.
So, good morning. I apologize for how I sound. It's a real pleasure to be here with all of you today, and it's always a pleasure to be back at Rice and a privilege to be back at Rice. My husband and I are both alums, and we love being here. The panelists this morning have covered a lot of ground. We've heard about the successes and promises of stem cell-based interventions, the importance of sound clinical trials, why and how the FDA is working to promote and protect the integrity of stem cell-based intervention research, and questions about the future of FDA activity in this area, regulatory models we might explore, marketing practices that promote the use of unproven stem cell treatments. I use that in scare quotes. I like stem cell-based interventions. The dangers that patients face in seeking these interventions, and the threat this practice poses to the future of research on stem cells. I want to turn our attention for a moment to the patients pursuing stem cell interventions, or the parents pursuing these interventions on behalf of their children, both in the United States and abroad. We should see patients' pursuit of unproven stem cell interventions as a form of communication by patients to the scientific and regulatory communities, and possibly even as a form of protest. The rhetoric of hope that clinics rely on and the false claims that some of them make are well documented, but we would be foolish to think that patients and parents pursuing unproven stem cell interventions are irrational and uninformed. There's something more going on, and we can learn something important from trying to understand their choices. They're telling us something in pursuing stem cell tourism. We should pay attention to what they're saying and find ways to incorporate that into policy planning. Doing so, I believe, is critical for developing an effective policy to address stem cell tourism. So what are they telling the scientific and the regulatory communities by pursuing unproven stem cell interventions? What can we learn? I think they're telling us at least 3 things. First, individuals and groups evaluate risks and benefits differently. A clinician might look at an intervention and think the risks are not justified. We don't know enough. We don't know whether it's safe. We don't know whether it's effective. For a patient, however, those same risks and benefits or potential benefits might look different. There's no single scale calibrated the same way by everyone that we can use to compare risks and benefits. There are risks and burdens that patients might find worthwhile given their overall circumstances. These might include the fact that they currently have a significantly diminished quality of life, or they expect to have significant limitations in the future. They might have a diagnosis that promises an early death. Patients aren't always risk takers, but in these cases, they have determined that they're willing to take risks, whatever they might be. Whether in hindsight they will agree is a different question, but we evaluate risks and benefits differently. And they're telling us something important in choosing to take on sometimes very significant risks. Second, they're telling us that they're not interested in vast data sets. The scientific and the regulatory communities often value significant amounts of data about safety and efficacy in groups overall. Patients are most interested in the effects on them. So Dr. Matthews mentioned people who have returned from pursuing stem cell tourism and been satisfied. In many cases, these are patients who have lived for a long time, hearing about statistics related to their diagnosis, their prognosis, and their treatment options. They want to know what's going to happen to them. It doesn't matter whether most people tolerate a drug well, for instance. If they don't, that's all that matters. If a particular patient doesn't do well with a treatment, it doesn't matter to them that it's effective in many cases. The opposite is true as well. Even if most people do not respond to an intervention, if they feel better, for whatever reason, they're satisfied. Their experience is the most important data point, perhaps even the only important data point. Significant amounts of data about what happens to other people do not matter to an individual patient, particularly when time is tight and the stakes are high. Third, they're telling us that they've waited long enough. What for a government agency might seem like a reasonable amount of time to initiate or conduct a trial or to approve an intervention may seem unreasonable to a patient. Some believe that they were misled by scientists who overpromised on what stem cell research could produce and when. In an effort to secure funds, the scientific community itself built hype around stem cell research. It now seeks to temper that hype with warnings that science takes time. This isn't the first time that patient groups have found themselves at odds with the scientific and regulatory communities, and it's not the first time they have communicated in what I would say, or even protested, in what I would say are finding ways to circumvent the clinical trials approach that Dr. Willerson talked about in order to access experimental interventions. So I'll just highlight 2 examples, and I think we can learn something important from these 2 lessons in thinking about the future of stem cell-based intervention research and policy. The first is activism around high-dose chemotherapy and autologous bone marrow transplantation for patients with advanced breast cancer. In the 1990s, it was thought that this combination, because it was successful in some other cancers, might be effective in helping patients with metastatic breast cancer who had sort of run out of options. It was very expensive. There was no evidence that it was safe and effective for advanced breast cancer. It carried significant risks. But there was no FDA barrier here. All the interventions were already proven. The drugs were available. But insurance companies were a barrier. They said, we're not going to pay for this. It's experimental. A number of patients and clinicians did not want to participate in the ongoing clinical trials on this intervention. They were afraid that they would be randomized to the standard of care, which was not highly effective for these patients. So they headed to court to demand coverage, and they succeeded. The clinical trials lagged while experimental interventions were provided, paid for by insurance companies or government third-party payers outside of clinical trials. Eventually, the trials were finished, and they showed no improvement in the survival of patients who had received the experimental intervention, and in some cases, they demonstrated that these patients lived less time than those who had been in the standard of care arm. The result was more harm than good to patients over a long period of time and extraordinary expense. The second example concerns HIV/AIDS patients in the 1980s who argued that the FDA's process for approving new drugs for this devastating disease, one that at the time routinely led to a very early death. Rather than wait around while people died, they began finding ways to import drugs that had been approved elsewhere or chemicals that were being used in other countries, and they distributed them in the U.S. They also started sharing drugs within clinical trials. This clearly compromised the integrity of the studies, but they believed that at least this way, everyone had a chance of getting something. So the people who were on placebo and the people who were taking the active agents were pooling their drugs. They didn't know who was who. They pooled their drugs and shared their pills. These activities did not go unnoticed. They gave the HIV/AIDS activists leverage with the FDA. They negotiated to formalize existing programs that gave people access to unapproved drugs and adopt new pathways for drug approval. You heard about some of these from Dr. Matthews. The activities led to the 1988 Fast Track Initiative, the 1990 Parallel Track Program, the 1992 decision to allow the use of surrogate endpoints in some studies. The results were a series of compromises that tried to protect the integrity of clinical research and facilitate access to new interventions earlier. This is not to say that the system that was created was perfect or that it met all of the interests of all of the stakeholders, but it shows that progress can be made in a way that acknowledges the competing vantage points of different stakeholders and that attempts to address their most critical interests without undermining other important goals. This is a worthwhile goal to adopt in thinking about the future conduct and regulation of stem cell-based intervention research. It's a goal that can be met only when these various interests and perspectives are represented fairly and acknowledged respectfully. All proposals for the regulation and conduct of stem cell-based intervention research should be measured in part against this question. Does the proposal reflect a compromise that meets the best interests and goals of different stakeholders so that we can protect the integrity of clinical research? So I'll end there. And
Many thanks to all four of our panelists for excellent, very enlightening presentations. So now this is the time when the audience gets to ask your questions of the panelists. There will be people with microphones going around, so if you want to ask a question, please raise your hand. We actually use the microphones because we record this, and people watching later do like to hear what the questions are. But I'll just begin. So go ahead and raise your hand so you can signal. Ms. Laymance has a microphone right here. But I'll just go ahead and begin by asking one question of Dr. Willerson. I was impressed by all the different advances that your lab is making and Texas Heart Institute is making. And we spend so much time, I end up thinking about personalized medicine and the advances in cancer, and it makes me think we're doing just as great progress in terms of heart disease. So I was wondering, in terms of these terrific advances like the mesenchymal cells, I'm a social scientist, so I've never had any experience in a lab. How difficult would it be for a lab in a foreign country to be able to try and replicate your treatments just by reading your scientific articles and going to see your work at conferences?
Dr. Ho, I don't recommend that anybody go to a foreign country to receive stem cells. We've heard a lot of important information about what can happen to people if they receive an intervention, in this case stem cells that have not been properly vetted, first for safety. Every stem cell study we've done began with studies in mice or rats or both, then progressed to pigs for safety and efficacy, and then humans. Unfortunately, or fortunately, the human is the right model to test any intervention. That's the first slide I presented, because it's pointed out what one finds in mice or rats or pigs or any other animal sometimes doesn't apply to humans. I don't agree that it's often doesn't apply, but sometimes it doesn't, or sometimes it's not safe. And so the FDA often requires, even after all of that testing, a run-in trial in a small number of humans to show that an intervention is safe. Then its efficacy is tested in the double-blind placebo-controlled trial. I try to discourage everybody who asks me not to go to Mexico or Korea or Vietnam or even some European countries to have these things done when I know that they've not been properly evaluated. We're in the infancy of this work. We're all somewhat impatient about it because I think all of you will recognize that the union of your father's sperm and your mother's egg, 2 cells, generated billions of stem cells in the human in a sac called a blastocyst. Those cells, I believe, were ordained by our Maker to be your heart, your kidney, your brain, your spirit, your function, your appearance, your behavior, everything about you. We're all walking stem cell products. The signals are genetic ones. So one has to decipher that genetic code, and you heard me talk about restoring aged cells to youthful ones with certain genes, converting skin cells, which don't want to be anything other than a skin cell, into your contracting heart muscle cells. It's all genetically regulated. But one's got to go through the right process, and you can be harmed by going to a place where somebody's doing this in their garage or somewhere else, and you have no idea about their training, their background, their capabilities, their results, anything. You know, humans are brave who are part of these clinical trials because there's some uncertainty in them about efficacy, unlikely to have harm. We haven't seen a single tumor from the stem cells we've used in some 500 patients. Not a single tumor, anybody. But we did a lot of testing to begin with to be sure we knew that. But you got to be very careful about it, and we can't get in too big a hurry. And there will be ways to repair organs by stimulating intrinsic regeneration, independent of the stem cell, by finding out our Maker's plan to prevent us from living forever or our organs from enlarging progressively. Somebody asked me one time if I was afraid of meeting my Maker. And I thought that was rather presumptuous, and I might meet my Maker to start with, but the second part was I told the person I was going to ask for forgiveness rather than permission.
Thank you, Dr. Willerson. Is there any questions? In the back. While the microphone's making its way over there, just a quick question to Dr. Frey-Vasconcells. It's horrifying what is going on in some of these trials that patients enroll that they can find on clinicaltrials.gov. I notice you mentioned that the FDA takes action against some of these groups. Is there any type of compensation that goes directly to the victim of a poor trial?
Of an actual trial, or?
Right. Right. You know, I sort of see you can shut the company down, and that's all good. Right. But these poor women who've lost their eyesight.
Right. No, actually not. There's no. I mean, the most they could do is sue the clinic where they got the injections. Now, I was actually surprised in that particular case. The state actually was able to shut. Well, I'll take that back. The clinic is still operating. They're doing injections for other things. They have stopped doing injections into the eye. It turns out that the person who did the injections was a nurse practitioner, and there actually was no physician overseeing these patients. None. So I think you really have to, if you're, I agree with Dr. Wilson. I don't condone any of these clinics. I think they're very dangerous. I think they are, I agree with what was said. I think they can severely inhibit science and truly getting effective therapies to patients. But I understand why patients go to them. If you do, ask a lot of questions. Really understand what you're getting into. There's some clear, signals that something's not right. And you just need to be aware of that. Now, I do know, like clinics, people that go to other countries, I have heard that there are physicians here in the U.S. that when you come back, they won't treat you. It's too much of a liability. And so you potentially need to think about when you come back, who's going to oversee your care. There's a lot of issues with this. And I don't know, I've not looked into this, but I'm not sure how insurance companies deal with this.
Interesting. There's a question in the back.
Hi, Dr. Willerson. Several years ago, you served on a committee for the Texas Medical Board when they were trying to maybe loosen the regulations on stem cell clinics in Texas. And you mentioned the idea of a registry. What are your thoughts on that right now, and what would a clinic that was part of that registry, what would their requirements be as far as like data collection, patient outcomes, and things of that nature? Thank you.
Thank you very much. Thank you for all the work you do on behalf of Texans for stem cells. We're all grateful. I do think there ought to be registries, and I think we could start with Texas so that these patients, individuals who are being treated with one thing or another, the person rendering that treatment needs to keep and submit for evaluation a record of whom he or she treated, for what purpose, what was used, and what the outcome was. And that we would have a regulatory agency, probably with the state and local representations, that would review these periodically. And that one would be able to identify fairly quickly things that were being done that shouldn't be done or that were harmful to people. So I sure would like to see that occur, and I'll keep lobbying for it.
I'm actually impressed that you recognize that there needs to be somebody to oversee the registries. Because I know there's been a number of cases, and I can name one. When I was part of the FDA sometimes will ask companies as part of their approval that they have to establish a registry. And I know this is a big deal in Europe right now. We actually had a product where we required a registry. And we recognized that we had approved it under accelerated approval. And so FDA has the right that if the clinical benefit is not demonstrated, they can withdraw the product from market. We actually took it to an advisory committee to ask whether this product should be removed from the market because we were seeing a lot of repeat surgeries. This was for an orthopedic indication. The company had this registry. We get to the panel, and the panel actually asked the company, well, what have you seen in the registry? And the company actually sat there and said, oh, we've never looked at that. So if you don't have someone overseeing the registry, what's the point of it? It's useless information. So I'm impressed that if Texas does a registry, make sure there is somebody overseeing that.
Someone has the microphone? Please.
I'm not a doctor or a scientist, but I'm a patient of a stem cell clinic. And more importantly, I'm a spouse of a patient from a stem cell clinic in Panama. I would have liked to have seen, and maybe you asked and didn't meet the criteria, a representative of one of those stem cell clinics from a foreign country or to be on the panel to give their side. My wife has MS and was diagnosed 10 years ago, have friends with MS. Hers is not real aggressive on a relative basis. Other friends have been very aggressive. They've tried all sorts of pharmaceuticals and were able to get some great relief and progress and healing, or symptomatically healing from the treatment. You're right, you've got to check those clinics out just like you would a doctor here in the U.S., or just like you would when you buy a car, or when you do spend any large purchase. There are, in my mind, obviously I'm not a scientist like I said, but the clinic we went to, Stem Cell Institute, seems to do the right thing and have a huge lab and do the right work. And they use umbilical cords from newborn infants. And long story short, we saw dramatic improvement in symptoms from my wife, things she could not do before the treatment, things she could do after the treatment. So I do think there is a lot of potential, as has been mentioned by the panel, of stem cell treatment. I don't know all the hurdles. I know there's a lot of hurdles getting through FDA. In my small mind, a lot of that is driven by the dollars of a drug company or an investment company backing a drug to get it through the trials. They will put the money together to go through the trials, knowing there's a large gain at the end when they get the patent and they get the drug. And then, as we're seeing now, they charge exorbitant prices. Some do, not all do, very few. The EpiPen may be an example of charging exorbitant prices to get their return on their investment. So there are good things about some stem cell treatment in foreign countries. I'm here to tell you that. And I know there's a lot of bad experiences, as we heard this morning, but there are some good things going on with stem cell treatment in foreign countries.
Can I address that? I think that's actually really interesting, and I've actually seen and heard stories like yours in the past. And I think really what I was trying to say is that I would like to see them to publish the data, them to release real data, them to do really systematic metrics. I would like them to pursue it in a scientific, and maybe perhaps they are, is that what you're suggesting, a scientific manner, but that's not what they're actually giving the public. And it's maybe this clinic is doing a great job and they found this little niche and they're able to help with the people with MS and they're doing really good work, but they're the only ones doing it. They're not sharing it with, you know, Dr. Willerson's group, the people in the medical center, so that we can expand on this, truly understand it, and then everybody with MS could actually have an impact from it. And so that's, I think, what my problem with the clinics that aren't doing the reporting the way we were hoping for is that they're not actually sharing it. So it might be one person, a small group of people having an impact instead of all of us as a public good. And I think that is how we approach biomedical research, and obviously clinical research. Dr. Willerson is an expert at it, so he can tell me whether I'm wrong, but that's how we approach it, is to try to get the biggest impact. It's not like something that we think is just gonna happen like personalized medicine with a small group because of a genetic profile. This is something we're hoping will have a larger impact, and they don't really give us that information. They're not telling us it's a special sauce, what they're actually putting in. They're not telling us how it's actually being run, and then they're also telling us how they're assessing the outcomes that your wife is seeing and whether or not they're recording it. I'm not sure if it's just you guys are recording it. And I know of a lot of patients who wrote their own little journal to tell us all of how their improvement has happened, and that's actually been interesting, but it's a one personalized case where we don't know what went in order to help her become better.
Undoubtedly, there are some parts of the world, places in the world where good work is being done. Certainly in large universities and cities all around the world, there's very good work being done. It's just that it's a problem for an individual person to hear about something in some of the foreign places and then go there without really any knowledge about what the risks are going to be. And our plea is that one be extremely careful with that. We're delighted to hear about your wife's experience.
There was a question here.
Is this right? He's done more of this than I have. Almost everything the gentleman just said before me is really what I was feeling, and we had a son that had a disease where his body produced tumors, and at the time he eventually passed away about 10 years ago. At one point in his disease, there was talk about the fact that stem cell might help him. And of course, as parents, we both, my husband and I both were, you do everything, we travel all around the world, you do everything you can to try to help him. And then there was one day that I realized that the way things are progressing, it will never help my son. And my husband continued to try to work toward the scientific approach to it, and I made the decision to do everything I could to make my son's life on a daily basis the best it could be. The frustration for me is in hearing your things that you said this morning and hearing this gentleman talk, I can relate to what he's saying. And I also understand what you're saying. And it seems to be there should be something, it may be this regulatory board, in the middle. Because in a way you're asking practitioners to be scientists. Practitioners are not scientists. And in my, because I see how things have to be done and how long it takes for things to be done. There's a sense of urgency when it's your life that's on the line or your child's life that's on the line. I think it would be, and it's almost that I get the sense that you understand that, but I know he really gets it. And there is a difference. There is a difference. And it seems like there should be some coming together in the middle. And me personally, if I was in that situation, I would say I do want every bit of education, information that I can get. And I want to know if 100 people did this, where they did it, and how many of it times it was successful. And just being fully informed that you go in and know, I might lose my eyesight. Some people may say, that's fine. So I think that is what you're talking about, is that people have the opportunity to be fully informed about what they're doing. But I think when there's so much potential in something to decrease suffering, not just to extend life or be able to walk faster, but to decrease suffering in people, there should be some hope, sense of urgency with that. Urgency with, doing it in the right way. I appreciate that. But putting so many restrictions on practitioners that they no longer can help people or give people the full opportunity to express, to know what they're doing, then it's kind of like, my gosh, it just
Thank you, ma'am.
Thank you.
And we're very sorry for your loss. Dr. Iltis, would you?
Let me, Dr. Ho, let me just answer it for a second. Of course, I understand that. I lost my own sister in November after she had pancreatic cancer, was irradiated, had a Whipple procedure, chemotherapy, appeared to be cured, developed breast cancer, metastatic, and failed 5 chemotherapy regimens at MD Anderson and Dana-Farber. And we were looking, of course, anywhere in the world where there'd be something new. But I wasn't going to send her somewhere just because somebody said they had something new. It would take much more than that. I'm a doctor. I treat these patients. I have over 3,000 patients. I watch them die. I watch them in misery. And our team's response to that, and this would be true of many, many physician scientists, is to find a way to help. It's one advantage that we have because we take care of people and we see what doesn't exist, and then we try to work to find it, to make it exist. I'm in a real hurry to stimulate cardiogenesis. There are children born with so-called hypoplastic left hearts. They have a diminutive left heart chamber, and they die early. They're operated on, but the operations are not very successful. I'm really hoping that knocking out this stop growth pathway will change that, or the stem cells. So we share that. I understand the motivation to look, personally and professionally, but you got to be careful.
Thank you. Dr. Iltis?
I think I really appreciate your comments. I think that's largely where sort of my comments were directed, at this idea that, look, people are telling us something important and we need to listen and we need to figure out how to work with that and how to incorporate that. One thing I just want to make sure we're clear on, when we talk about stem cell tourism, we're not just talking about foreign clinics. We have clinics in the U.S. that are doing the same things, and some of the examples were from that. So I think that's just important, not to think that we've got it right and it's everybody else who's doing it wrong. So I do think we need to take that very seriously. There are real reasons why people are going to clinics pursue a whole number of different interventions. I have a student who's from China. We were talking about stem cell tourism in my class, and he said, that's what my aunt and uncle did. I have a cousin with Down syndrome. And they spent, and he told me, the equivalent of $180,000 on an intervention in China, which did not have any effect. And fortunately, I said, well, fortunately, it didn't have any effect. Fortunately, your cousin is still healthy, because it could have been terrible. But I do think we need to listen. I think we need to understand there's real reasons why people do these things. And in many cases, it's not a matter of being uninformed. One of the real difficulties is that many of these clinics are not willing to participate in what in any other setting would be standard medical, clinical, or clinical research practices. So the unwillingness to be specific about what's being done, to be systematic about recording it, about reporting it, about allowing others to attempt to reproduce it, right? That's an important part of developing effective treatments, is knowing that others can reproduce what you have done. And in order to participate in that process, we have to have clinics and practitioners, clinical researchers. So often clinicians are, in fact, also researchers. They're clinical researchers to help us develop, over the long term, safe and effective treatments. So if they're doing things that are working well, participate in the system, participate in the process of reporting, going to scientific conferences, and truly giving details about what's being done, and to be systematic about it so that other people can try it and try to reproduce it. That's why we have so much of what we have today in healthcare. That's where it came from. And so we need to continue to participate in that system. And I think we really have to develop a regulatory model that allows us to do that. We can't ignore what people are telling us.
Thanks very much. And I'd like to thank all of our panelists for a wonderful presentations and a discussion. I'm sorry I have to wrap up right at 9:30. I actually have a class I need to go teach upstairs, and I know Dr. Willerson has another obligation he has to meet. My guess is the other panelists would be willing to stay and answer questions one-on-one if you still have some matters you'd like to discuss. Thanks so much for coming.